Good afternoon, everyone, and welcome to our D's Distinguished Lecture. It's hard for me to believe that we started this series back in 2014, over 12 years ago. And initially, the logic behind this series was to make all of the faculty in the different departments of the school familiar with our research. And then we decided to branch out and invite more of the community so that they can learn about what is happening here at UCI. I am particularly excited by today's speaker because, as someone who occasionally relies on their GPS to navigate, I'm really excited that we're going to hear from an expert who studies human spatial learning and memory. So that's a great pleasure for me to introduce my colleague, Liz Chrastil, who is from the Department of Neurobiology and Behavior, and she also holds an additional appointment in the Department of Cognitive Sciences and is also a fellow of the Center for the Neurobiology of Learning and Memory. Let me just give you a little bit of background about Liz. She began her career at our sister school, UC Santa Barbara, but was drawn to UC Irvine about 7 or 8 years ago, mainly because of the strengths in neuroscience and learning and memory and aging here. And as I indicated, she studies spatial memory, how the brain supports navigation. Her work is truly fascinating. It's been featured very widely in a lot of the press, particularly because she studies how immersive virtual reality and uses neuroimaging approaches to better understand how we build mental maps of our surroundings. Her research has a lot of implications, not only for memory, but for aging and Alzheimer's disease as well. In addition, she's also participated in as a research subject, and while pregnant, her brain was imaged throughout her pregnancy. So that's tremendous dedication to understand how neural changes unfold in the maternal brain throughout gestation. As you all know, pregnancy is a period of profound hormonal and physiological changes experienced by millions of women annually, but yet very little is known as to what happens to the human brain when during that time period. And this study has led to a publication in 2024 that's already been cited over 120 times and has been assessed over 325,000 times online. So that's pretty remarkable. She is a very interesting young woman who is also an avid dancer and recently taught a class on how to dance at weddings. And so she is, I think, she holds the distinction of being the youngest Dean's Distinguished Lecturer. So let's welcome Liz Chrastil.
Well, thank you so much. Thank you for inviting me. It's such an honor to be part of this lecture series. Oh, where'd the clicker go? Oh, there it is. Okay. Thank you so much. It's truly an honor to be asked to give this fantastic lecture. I've seen so many really fantastic researchers here, and so I'm really honored and delighted to be able to share some of my work with you all. So my work is on human spatial navigation, and I just want to talk a little bit about one aspect of things that we're going to cover today. So this is quite a mouthful of a title. We're looking at individual differences in navigation ability. And I just want to start by thinking about how we're all different, right? So we all know we're a unique person, but I want to give you just an example to start thinking about why some people might be better at spatial navigation. So I'm going to give you two people and tell you a little bit about them, and you can sort of think about whether you think person one or person two would be better at navigating. So person one is a 27-year-old man. He's good at mental rotation, which is this ability to sort of think about an object and see it from a different angle and understand what it might look like from the other side. He plays a lot of video games. But he takes the same routes to work every day and maybe uses his GPS a lot. And so he takes the same route home and from home to work. He has really good myelination of his brain. And now myelin is the sort of fatty tissue that coats our neurons, and it sort of helps the electrical signal get processed. So it helps you transmit the electrical signal. So having good myelination really helps you with sort of brain communication. But he's chronically stressed. He has a really demanding job, and maybe he has some financial troubles as well. Okay, so person 2 is a 50-year-old woman. And she has a great working memory, and that's our ability to sort of keep track of like numbers, for example. So like if you get a code on your phone and have to put it into your two-factor authentication, that sort of memory. She has great pattern separation, and that's a memory ability to keep track and separate, say, for example, what you had for breakfast and lunch yesterday from what you had for breakfast and lunch today. These are very similar memories, but you can keep them separate. She likes to run and hike. She has a larger hippocampus, which is a really important part of our brain, which is the sort of most important brain region for learning and memory and also for spatial navigation. But she gets plenty of sleep. Oh, that's nice. Getting lots of rest. Most of it do that. But she also has a family history of Alzheimer's disease. So there's a lot of things that go into people, right? And I sort of mix them all up. So maybe it's not obvious which one of these people might be a better navigator from these things. And not many of these things have to do with navigation. But they're all different aspects of our lives and different things that might be important for how we behave in the world and how well we are able to do things and how we're able to navigate. So there's lots of different factors that go into ourselves. It's not just cognitive abilities, but there's all kinds of different things that really help us perform a really complete profile of everyone and understand who we could be. So why are individual differences important to study? Well, first of all, it's just fun, right? It's fun and interesting. And I think that's something that's really important to think about in science, that we all want to know what makes us unique, what makes us different from other people, what makes us special. And, we think about our friends and, I think we all know somebody, and you don't have to identify yourselves, but some people who are really bad at navigating and you use your GPS every time to get home. And we also know people who are really great at navigating, right? Who can get around very, very easily. It can be at place once and they can just completely repeat it, right? So it's just fun. And one thing that I really enjoy about my work is that when I talk to folks, everyone has a story, right? So it's very relatable. People, yes, oh yeah, like I'm always navigating, my partner's driving because they can't find their way, things like that. So we all know about this and it's fun. But we also want to help people too. And so thinking about from a more public health standpoint, we're also interested in precision medicine, right? So we want to understand each individual's cognitive profile because we want to be able to say, well, this person might be at a higher risk for a disorder, right? And so we're particularly interested in Alzheimer's disease. That's the one that's most relevant to my work, but also things like depression or OCD or other aspects of mental health that could be relevant. So we want to know who might be at higher risk. We might want to see who might be resilient, who might have a lot of risk factors, but still not develop something. And then we want to be able to have targeted preventions and treatments, right? So if I'm at risk and I have a certain profile, I want to be able to say, this is what I need to do to help prevent Alzheimer's disease. Or if I have started to develop something, here's how I can treat it, which might be different from somebody else because you all have slightly different factors. So overall, we're trying to characterize each individual's sort of cognitive, but overall personal profile so we can really have a sense for who's likely to have problems. So how do we test navigation in the brain? So just what do we do in my lab to do these questions? I want to start with just showing you some of the things. We use immersive virtual reality. So this is an example of one of our maze environments. That's what the participant sees, and this is what's actually happening. So they're walking around the lab. It's just a big empty room, and they see virtual environments. So we can test them in different ways. We can make more complicated environments or less complicated environments. Sometimes we make a virtual desert so they can't see anything really, and we just ask them to walk around in this virtual desert for different reasons. And then we use magnetic resonance imaging. And so we have our fiber center here, which is our imaging center. And so we can look at things like anatomical scans. We can look at gray matter and white matter. And on these images, the gray matter is sort of gray. These are the main cell bodies, and our axons are the white matter. And we also have more fun and colorful ways of looking at white matter with diffusion imaging. We also do functional stuff too. So we have lots of ways to test this in combined. And then we can also do not the walking around versions, but we can do sort of like video game versions of our virtual environments in the scanners because people can't walk around in there. Okay, so let's think about then factors of navigation ability. So we can think again, there's lots of different factors that might be involved. And first factor I want to talk about is behavior. And that can encompass a number of things. I can talk about sort of cognitive abilities, but also like how we act in the world, how we maybe learn, how we explore and get around. We also have our life experiences, right? So perhaps you like to hike and run, and you like to maps. Maybe you did some art, other things like that. There might be lots of reasons of the things that we've done in our lifetimes that can help train us to be better at navigating. And then, of course, we have our brains, right? So that kind of underlies a lot of this work. And our brains support all of this information in different ways. And we have structure and function, and I'll go into talk about that more. We also have our age. We can't really do much about that one, but that's one where, things might be very different across the lifespan for different people. And we have sex and gender effects, which can also interact at all these points. So these are sort of our core things that we can't do too much about because they're sort of in our past, but we also have things that we have a little more action on: sleep, exercise, diet, stress, and the environment that we're moving around. So we have interactions, things might be different in different environments. And so this core of sort of like our intrinsic stuff about ourselves, but then all these external factors as well that are all play a role. And so they all interact with each other. The internal stuff interacts with the external stuff. And so I'm going to focus today on these core factors, but I also do some work with these other things as well. And it's important to think about that because we have all these factors, but navigation has lots of things, right? So there's many facets to navigation. It's not a single entity necessarily. And so, for example, knowing where you are and seeing a landmark is one type of spatial ability. So just being able to put yourself in a place recognition, seeing the anteater statue, might be very different, for example, than following a route and being able to know a series of left and right turns and just being able to say, turn left here, turn right there. And that might be a little bit different from something like reading a subway map, where you have to kind of figure out, okay, I take the green line 4 stops, and I change to the yellow line, and I go 3 stops. And that provides some different types of information. And that might be very different from perhaps what we do in our sort of day-to-day lives, getting around. And also could be very different, for example, even just being able to say like, okay, we know to get to downtown Los Angeles, we take the 405 to the 110, provided the traffic's okay, and we can get there. But if I ask you to just take a straight line, it's actually something slightly different, and you could do something like that. And then finally, that might be all very different from like walking in a desert and just saying like, I'm going to walk in a straight line and then repeat that distance. Or close your eyes and walk in a straight line. It's actually hilarious. People can't really do that very well. But stuff like that, those are all parts of and aspects of navigation that could be important for us. So let's start talking a little bit about these different factors. We'll start with our behavior. And that's sort of my core. I love the behavior. So we'll first start about talking about cognition and different aspects of cognition. So I mentioned before that there might be other cognitive abilities that might be important for our navigation. And so this is our sort of like working model of how we think it all fits together. And I'm not going to go through all the details of it, but it's basically, a theoretical model of how these different abilities might connect to navigation. I think what's important here is that we have some factors on the left, and they each tested with multiple tasks. So the pointers, these factors are all involved, and they might load onto different ways to these different navigational abilities, and includes things like personality as well. So there's lots of things, small scale spatial ability like mental rotation, but also that sort of walking in a desert environment all build up together. And so we can test how these cognitive factors map onto our navigation performance. And so this is a big study that we just completed data collection on. And this is work in collaboration with Mary Hegarty at UC Santa Barbara, but also Craig Stark and Jeff Kritchmar here at UCI. And we've been working with this. We tested a lot of people. We tested over 250 people across 2 sites for 10 to 12 hours of behavioral testing. So this represents between 5,000 and 6,000 hours of data collection from our staff. So they did an amazing job doing this so that we can really, again, develop this full cognitive profile of people. And we had these 11 different virtual reality tests. So we had some in that sort of virtual desert or sort of just like a virtual hallway. Some were in these more simple, like a circle environment. Some more in the maze type and things, and sort of a variety. And they're all in this walking, some were walking and some were desktop VR. And then half of those folks, in addition, got an MRI scan of basic structure and function. And so I'll be pulling some of that out. And so we're still working our way through a lot of this data. So we don't have too many results here, but I just want to point out that we have, a first pass, and this is, again, I'm not going to go into all the details about this one, but basically we find that there's some things that are important. So our model was a little different than what we thought. The spatial visualization kind of loads in together. So this is things like mental rotation, ability to take a different perspective from where you are, and being able to say, like, okay, if I go down the street this way, things are on my right. If I come this way, things are on my left. All kind of load together. Learning and memory loads separately, and so does path integration. That's the sort of desert environment. So we have a couple different things that are independently loading onto navigation ability. And navigation is actually related to these several other cognitive abilities, but it's also a bit unique as well. So it's not fully explained by anything else. It's not like navigation is all learning and memory or all working memory. It's really its own thing. This is being led by Mitch over at Santa Barbara. We also, again, we have like tons of papers that we're working on right now with this one, but I just want to give you sort of one result that we've come up with so far. And that's looking at the activities that are related to performance. So we talked about what might be useful for you in your lifetime, what kinds of experiences might be the best sort of thing to help you be better at navigating. And what we found was video games. It was really video games. So of all these things, we did a whole bunch of different questions, and video games was related to navigation performance for both our desktop environments and our immersive environments. And that's really important because it doesn't mean it was just the controller. So if it's just the desktop environments, it might just be the controller. But you're walking around in these complicated environments, and still video games are helpful. It's also a little bit more so in large scale spaces. The other thing that came up was that GPS use is related to worse navigation performance in our performance. Now, there's a couple things here. So one, this is a young adult study. So these are only 18 to like 30-year-olds. So these are folks who have grown up with GPS and who grew up playing video games. So it's going to be really interesting to see what happens when we compare this to, say, 70-year-olds who didn't grow up with either of those things. It may be very different. So we're going to see what happens when we do that. The other thing to note is, especially for like GPS use, it's hard to know the causation, right? The direction of this, right? We know they're related to each other, but we don't know if people who are bad at navigating use their GPS more because they need to get around, or if it's sort of, you start to lose something. So that's something we're going to investigate more. All right, so in addition to all these other cognitive factors that might be involved in our navigation abilities, we also have things that we do. And so like how we act in the world, and that's another type of behavior. And I'm going to talk specifically about exploration, like how do we learn our environment? What is helpful in learning? So I'm going to share with you a study that we conducted, which is in fMRI, and we had I'm going to take this away for a second. It's a maze environment, so this is an overhead view of the environment. So there's some alcoves here with objects in them and some paintings on the wall. And so now I'll show you what's happened. So at each intersection, the person just presses buttons to get left or right. So we try to make it really simple controller, just left, right, or straight. And this is an example of one person moving around. This is a test trial. So they first explore for about 16 minutes. And we just say, find all the objects and learn their locations. So you don't have to do anything too crazy. You don't give them any particular task, but they just need to learn where everything is. And so from that, we can really figure out whether people are, how people are moving around to learn, right? So we're interested in the learning process. Well, how are people learning this? And so then at the test phase, they're asked to go from one object to another, but we remove all the objects. We turn them into these red balls. So it is possible to get everything wrong, and people can and do that all the time. So they actually, a number of people get 0 right, but people get 100% right as well. So we can really see the range of individual differences here. So it's really nice with that. So our go back. So our range of performance here ranges basically from 0%, this is we call chance, all the way to 100%, and sort of everything in between. So we can really leverage these individual differences to understand then what people are doing. And so we'll see this task a couple of different times. I'll be using this task a number of ways today, so just important to keep that in your mind. So first we looked at this exploration. This is work from my former student, Erica Ward. And, and we were like, so what happens in exploration? How can we tell whether people are going to do well at this task? So one thing that we noticed is that finding all the objects faster was related to better performance. So if you spent like 200 moves to find all the objects, you were going to do really poorly. And if you found them pretty quickly, you did really well. So pretty strong correlation there. We also noticed that even exploration was really important. So if you explored all the different spaces about the same amount, then you would do much better at the task than if you just sort of circled around over and over again. And so just returning to that object exploration, we can zoom in on this a little bit. So here's object 5. Doesn't matter which object it was, just the 5th object that you found. We can already start to see differences between the group who was at the bottom, the worst performing people, and the people who were at the top. And it sort of gets worse and worse. The difference gets bigger and bigger as you go along. So object 5 is about 20 to 30 moves, and that's about like 3 to 5 minutes. So if we watch you for about 3 to 5 minutes, we can tell whether you're going to do poorly in this task or pretty well. We sort of developed that into a principal components analysis, and we found out sort of one component was just like we call quantity. So how many buttons did you press? How far did you travel? How many objects did you visit? Things like that. And you'd think seeing more stuff and traveling more would be better, right? You'd have more experience. But we also found a quality measure, and that's including things like this moves to find objects. It includes things like the evenness that I mentioned before, but only the quality measure was related to performance. And again, this isn't our healthy young adults. So really it was about that even exploration, finding things early, that was super related to performance. We've also looked in some non-spatial environments. So we also have a whole line of research that actually is looking a little bit more abstractly at things. So we have a non-spatial navigation task. So it's a very different, but we can also look at exploration in this task as well. And here we can look at, see what the participant is seeing. So here, people just see like pairs of objects, 2 pairs or 3 or 4 objects, and they just say, choose one. And then when they choose one, like some new stuff shows up. And they're kind of like, what am I doing? And you're just choosing stuff and moving around. But unbeknownst to them, there's actually, it's like a structure. It's like a maze, but they just don't see the maze structure. And so we have these different environments here that people can move around. And then at the end, we ask them, just like, get the orange on the screen. And they can press the buttons and get to the orange. So it's a non-spatial task, but it's basically the same idea. And here we have several different environmental structures. We have some that are really random, something that's like more grid-like, like New York City, something that's got these like we call communities. It has these little nodes together, little clusters, and some that have these little bridges in between. They're very similar to the other one, but they just have like a little bridge between communities. But these different structures seem to make a lot of difference, actually. So one thing is that performance actually varies by environmental structure. So there's not that many people who are actually great at all of these. They're not just like some people do wonderfully overall. It's like some people do really well in the grid environment, some people do really well in the bridges environment. And so there's a little bit of you involved. So for example, one thing we noticed that the number of bridge visitations is related to accuracy. So even though it's just a very simple location with just like one thing there, being able to connect there it helps you do better. So something about how you explored and visiting those bridges more often was really helpful. The other thing is that we can connect that back to some of our other cognitive abilities. And we found also that when you explore, some people explore for information gain. So they're finding, it's basically this evenness of exploration, right? They're finding places they haven't been before and they're trying to explore for information gain. And we found that pattern separation, which is the thing I mentioned before, that's about being able to distinguish lunch yesterday from lunch today, is actually related to how well you do this information gain in all different environmental structures. So there's something about this ability to keep track of different patterns and keep memories separate that's also important for how you explore. Okay, so just to sum up a little bit about this behavior section, we found we have a large individual differences study that's ongoing. There'll be watch this space. We'll have a whole bunch of more things coming out, but we just have our first analyses from here. So we found that spatial visualization, episodic memory, and path integration are related to navigation ability. That video games and GPS use were also related to performance, but that even and complete exploration geared towards novelty and new information is better. But one thing that's important to note is that there's not an ideal exploration for all people. So if I give you an exploration pattern and I say, go around in this way, it's not going to necessarily make you do really well at this task. And so we've done that. We have guided, active, and passive people, and they're not necessarily related. So there's something that people are bringing to the table, right? So when you're exploring, you're finding information for you, the information that you don't know, things that you are trying to find out, which might not be the same thing that someone else is trying to find out. And so, again, we're trying to, there's this strong relationship with the individual and what you're bringing to the table. And the thing that I think you're bringing to the table is your brain. And so that's our next stop, is thinking about how the brain might be related here. So we're going to start with structure. When I talk about structure, I mean things like how big different parts of your brain are, how well they're connected in different ways. And so our brain structure, we're going to start with hippocampus volume. So the hippocampus is this fun little region of our brain that's super powerful and it's important for learning and memory. So if you don't have a hippocampus, you can't form new memories. So it's super important. You're going to have trouble navigating as well. So you might think bigger is better. So let's have a bigger hippocampus and that will really help us with doing better at this. So we investigated this. So my student Alina started looking at this with that first maze task that I showed you, the one where we did in the scanner. So she did a bunch of, sort of corrected for different things like age, sex, and total intracranial volume. And she found some relationships. So she found that accuracy was correlated with some subregions of the hippocampus. So for those who are interested, dentate gyrus, CA1 and 2A, CA2, 3, as well as total hippocampal volume. But what we will notice here, this is our total hippocampal volume image. And this dotted line is the overall trend, but then she also separated it out between males and females. And so here, it's actually kind of in the opposite direction. So if you've ever heard of Simpson's paradox from your statistics class, this is actually a great example of it. It's like the perfect example. So this is where basically the overall trend is in one direction, but when we split it up, it disappears or goes in the opposite way. So when we ask a slightly different question about whether hippocampal volume predicts navigation ability, it doesn't predict particularly well. That sex is actually the stronger predictor of navigation performance. But it is related, but just not as necessarily like the strongest thing. So we also can look at different aspects of structure. So I mentioned that we have gray matter and white matter. And so hippocampal volume would be a gray matter measure. And so that's looking at how big, that volume is. But our white matter is that connections, those long range fiber tracts that connect different parts of the brain and let us have that functional communication between brain regions. And so you can look at how well that white matter is myelinated or different properties of the white matter. And so again, we looked at that maze task and said, okay, let's use diffusion-weighted imaging, which is a way of measuring white matter. And again, it's the axons, the output of these things. And we're particularly interested in the axons that come out of the hippocampus. So the white matter tracts that are then taken from the hippocampus and communicating with the rest of the brain. So for example, prefrontal cortex or other brain regions that might be important. And so we'll take a look at that. So diffusion-weighted imaging just uses water diffusion, actually, and seeing how well it diffuses in your brain. And so here we found that these hippocampal outputs, they're particularly called cingulum bundle and fornix, which are these sort of ones like going like across the front and back. But motor regions, including the cerebellum, were also correlated with performance. So we have sort of both these hippocampal outputs that are related to performance and some of our motor regions. So we see the limbic structures, which are those hippocampal areas, are important for this task to do well. So hippocampus is still important, but maybe just not bigger. Okay, so I'm going to switch gears just a little bit to talk about how some other ways that we can figure out whether people are going to do well at these types of navigation tasks. So one thing we're doing with that big new data set is using some machine learning to try to figure out if we can predict performance. And so my student Marjan has been working on this in different ways. And she's been started with just a mental rotation task. So it's not necessarily a big scale navigation, but something really simple like just can you tell whether or not which one of these is, whether these are the same or different as they're like differently rotated. And again, this might be important for navigation. And here she did something really simple. So normally with machine learning, you might just say, okay, here's the percent that they did. They got 85% right. Here's person 1. Here's person 2. They got 75% right. Here's person 3. They got 20% right. But instead, she asked a slightly different question. So she asked, here's 2 images. Which performed better? Oops. Which person performed better? So it's a little different. It's a comparison. So now we say person 1 or person 2. And so we can do this throughout all these people. So instead of having 8 different people and we have 8 different values, 75%, 80%, here we have 28 comparisons with 8 people. So it amplifies our data quite a bit. And then when you scale it up to the 108 people that we had in this study, we now have almost 5,000 data points. So this is a way of amplifying our data for machine learning. And machine learning really requires big data, right? So it really has a lot of catastrophic failures if you have smaller samples, but with larger samples, you're able to do something for real. And so what we then found is then by doing that comparison with this mental rotation task, she can predict like with really great accuracy, how well people are going to do in this task. And this is just taking a basic anatomical image of their brain. So you can also put a lot of other stuff in, right? So this is just this sort of gray matter, white matter image. But you can put all kinds of other things in. You could start putting in those diffusion images. You could put other types of things. You could put functional images in as well to make it even better. And so then the next step is to say, well, what parts of the brain are important for this? And so again, we're looking at this mental rotation. So when we put the model in, we can start taking chunks out. And so when you take a chunk of the brain out and the model doesn't do very well, then it means that part was important. So if you take it out and the model's fine, then you're like, well, it didn't really matter. So through this like backward elimination process, we can start to see the areas that were important for this mental rotation process. Some areas in the frontal cortex, some of our more parietal areas as well. And so we've seen different areas for different tasks and are trying to find out more about this. So I think this is a really exciting way of thinking about how we might do some of this work in the future. So in addition to brain structure, we also have function. So that was all just like structural stuff, like how big is our brain? What's a picture look like? But when we think about our brains, usually we think about function. What's happening? How are the neurons firing? How are they firing in communication with each other? And so that's a really exciting thing to start thinking about as well. And so we're going back to that big navigation task. Again, Eric has done this work, and we could start thinking about communities of brain regions. And so a community of brain regions are just groups of brain regions that have sort of a functional cohesion. So they tend to have correlated activity or cohesive activity. They just basically work together in different ways. And so we start by breaking up the brain into some parcellations. So we break it up into about 215 little regions. And then we do a correlation matrix. And so we basically just say like, I think it's a cohesion basic, but basically we see like which parts of the brain are having correlated activity with other parts of the brain. And then from there, we can run a couple algorithms that will break this up into about, 3 to 12 communities at any given time point. And that can help us see sort of how these different units work together. And then critically, rather than just doing this at one time point or just looking at this overall, we can actually do this over time. So we can start to see whether these communities change over time as we go through. And just from the start, we see that the people who did worse at this task have fewer communities. So they just break up into fewer number of subsets than people who do well at this task. So just like the number is smaller, but then they also sort of act differently too. So we can also look at this dynamics. So we're interested in thinking about how these brain communities might change over time. Are they staying the same? Are different nodes, which are these different brain regions, are they hopping from one community to another? So we can think about that. So here we wanted to look at reconfigurations of the community structure. So again, changing from one place to another. And so we're interested in something called flexibility, which is the time a node changes communities overall. So here, for example, is a very inflexible node. It stays in the same community, this sort of light green community all the time. But here we have a flexible one. It switches from the red to the yellow to the red to the light green. So it's moving around at each time point. And this flexibility has been associated with communication between different brain regions. It's been associated with learning. And so understanding this flexibility might be super important. But not all flexibilities are the same. We can break the flexibility up into two different types of flexibility. One is called a disjoint change, where one brain region just switches to a different community by itself. And so it's all on its own, and it just moves over and doesn't do anything, or it forms a new community. You can also have cohesive changes where more than one move apart. So you might think these disjoint ones might be noise or something, but they actually turn out to be really important. And so when we look at correlations with performance, only disjointedness was correlated with performance. We see that overall flexibility or cohesive changes were not, but these independent reconfigurations are really what's driving performance. And this was true in the exploration phase. While people were learning, this disjointedness was especially important for learning, but not so much during the test phase. So we see starting to see some ideas about how change over time might be useful. So just to summarize here, we saw structural relationships with navigation performance in terms of hippocampal volume and white matter integrity. We are using machine learning to do multimodal brain imaging to predict performance. And then we see some functional dynamics. So now we're going to sort of roll into our final couple of sections, and we're going to talk about age. And I know it's not fun to talk about age, but why is it important? And this is where I think we connect a lot of what the work I'm doing with sort of some broader health issues. So there has been a large overlap between the brain regions earliest affected by Alzheimer's disease and those that are important for navigation. So the earliest areas affected by Alzheimer's disease are entorhinal cortex, hippocampus, and retrosplenial cortex, and some of parietal cortex. And if you ask me what are the three most important brain regions for navigation, I'd say entorhinal cortex, hippocampus, and retrosplenial cortex. So we see the exact same brain regions that are important for and hit earliest by Alzheimer's disease with those that are important for navigation. And so one of the things that we're interested in is saying, well, can we use navigation as an early cognitive marker? But perhaps more importantly is saying, like, are you seeing cognitive deficits? How can we see the trajectory of cognitive deficits at these early stages? Okay, so as I mentioned, navigation is this early marker. Even for people like decades ahead of time have seen some differences in their brains in the entorhinal cortex and other areas. So healthy young adults who otherwise seem to be fine have shown some differences in their navigation and in their brain. And then another really important thing that I'll talk about in just a few minutes, but is that women have twice the dementia risk than men. We don't necessarily know all the reasons for that. And that's something I'm interested in finding out more, but we do know that that's the case. And as I mentioned, as you've already seen, there are some pretty big sex differences in navigation that have been frequently observed. Men tend to do better at a lot of these navigation tasks, but not all. So it's interesting to see where there are big sex differences and where they're actually pretty minimized. So we can see a lot of these differences. And finally, sex hormones influence the organization of the hippocampus. There's a ton of estrogen receptors on the hippocampus and in other areas of the brain that are really important for this. So that really suggests that there might be this link. And so when we think about the possibility of understanding why women in particular might be vulnerable to Alzheimer's disease, that's something that we want to understand more, especially in the context of navigation. So again, characterizing each person's cognitive profile so that we can understand that more. Okay, so I'm just going to talk about what we're doing now. I don't have any results here to share, but we're doing some work with Alzheimer's disease and exploration behaviors. My former postdoc, Isaac, who's developed the supermarket task. So it's a really naturalistic task where people can sort of walk around and do something that they know, like groceries, and see whether or not we can have exploration behavior is another early marker. So whether, again, how people are moving around, how they're exploring, can somehow provide some sort of clue. We're also doing some sleep and navigation in older adults. This is with Eitan Sheckman's group, and thinking about, this is not necessarily Alzheimer's related, but just thinking about how we can reinstate spatial memories and help strengthen navigational memories for older adults, again, with sort of a long-term eye and thinking about Alzheimer's. And then we can also look at individual differences and Alzheimer's disease more longitudinally. So I think one thing that comes up a lot when I talk about this is like, okay, I'm 20 and I'm bad at navigating. So am I going to get Alzheimer's disease, right? Is that something we don't know? So we don't know if it's like something really early on is going to be like critical and just like telling if you're bad at navigating when you're 20. Or if it's more like a particular aspect of navigation. So that's something we really need to figure out is like what aspects are going to be important, right? So again, navigation is multifaceted. So some aspects are going to be maybe important and informative and others might not be. It could also be that there's a particular change, right? There might be a change at a particular time point that's critical. So what we don't know is whether it's like a baseline or a change. So we're going to find that out. Okay. So when I talk about age, though, one of the things that I think is going to be important is these earliest stages of aging. We're not really focused on a lot of like super older adults, but we're actually just looking at the very earliest, which is like 45 to 55 or midlife. And so again, not very old. I know I say this, I'm in that realm, but we're just really thinking about, the earliest things that might happen. And we know aging can start happening even, in your 20s and 30s. But so like what's going on in midlife? And so we're going to go back to that wayfinding task. Just to remind you, can see the video again. But we have, in addition to our younger adults, we've also had midlife adults do this. So we've had midlife men and women do this task as well. And so just to go back to the behavior, our young adults performed okay. We see this spread. So our men did pretty well, our young women did okay, but our midlife folks did quite poorly overall. They had a reduction in performance. In particular, men kind of dropped off, and they didn't do as well in the midlife years. But let's look at that exploration behavior again. So this is Weizsäck's work, looking at the exploration behavior. So we noticed that, so although our young people did better than our midlife people, our midlife folks actually spent more time at the objects. They actually saw the objects more often. They spent more time. They spent less time in the hallways. But that didn't help them, obviously. So seeing objects or more time was not necessarily beneficial, but really exploring in a different way is more helpful. And then we did those same principal components. We looked at the quality score and the quantity score. In our young adults, it was just quality, but in our midlife adults, both quantity and quality was important. So there might be something a little different about how you might explore that might be useful in a midlife. Then we can connect this with the brain a little bit. So this is work of my student, Daniela Cusillo, who was looking at the white matter tracks and seeing how we could relate those white matter tracks. So we have the white matter integrity. In this case, in our midlife group, we only saw white matter in our sort of motor tracks, our cortical spinal and other motor tracks was related to performance. Whereas if you recall, our young adults had those same motor tracks, but a whole bunch of other things and that were in these sort of association fibers and these more limbic tracks that were important for hippocampus. So we saw some differences there. So our midlife folks are not really using those hippocampal outputs to the same degree. Okay, so one reason that we're interested in midlife is because that is when women undergo menopause. So I mentioned that there's these big sex differences, and I mentioned that there's a lot of work comparing that women have this higher risk. And one idea is that what happens during menopause might be important. So we can look at this wayfinding task again, and then we can now separate this white matter tracks here. So we can first of all just separate between men and women. So in addition to the overall findings of these motor tracks, we see that men actually have negative correlations in the fornix, which is that hippocampal output. So actually having a better fornix essentially makes you do worse at this task, is related to worse performance. It's kind of unusual and unexpected. But women, so both men and women had sensory motor areas, but premenopausal women had the opposite. So premenopausal women did what we were expecting, which is having greater white matter integrity was related to better performance. So we see in this case that you can have sort of similar behavior. They actually performed similarly well, but they have different brain support. So you can have, men doing one thing and having sort of different ways that the brain is supporting, and women having a completely different way that their brain is supporting performance here. One more task that I want to just mention briefly is called the dual solution paradigm. And here we're not actually asking about how well you do this task, about how well you find things. We're asking a strategy question. So how we get at strategies is we first have people learn this route through the maze, and they repeat it multiple times. So they learn it really well. And then we ask them to go from one object to another. For example, if we asked you to go from here to here, you could take that route again, but it'd be kind of a long time. Or you could just take a shortcut and go straight through. Right? So what we want to know is whether people take the route they know or they take a shortcut. So it's really about like their choices here. And so here we saw that our young males tended to take more shortcuts, more than anyone else. So our midlife folks didn't take too many shortcuts, and our young women didn't take that many shortcuts overall. But when we again look into the brain, there's a whole lot of really interesting age differences I'm not going to talk about here just for time, but we see that men and women both have motor tracks related to taking fewer shortcuts. So again, motor related now to fewer shortcuts and taking the learned route, which make sense if you're thinking about sort of a motor plan. But here in our women, we saw that both pre and postmenopausal women had this fornix was related to more shortcuts. So we do see something about hippocampus related here. It's a little complicated, but what's perhaps a little bit more confusing is that premenopausal women take more shortcuts than these peri and postmenopausal women. So even though they have the same brain support, they are having differing behavior. So they're having different ways, different amount of times that they take shortcuts, but we again see the same relationship. So the more shortcuts you take, the better your hippocampus is working. Okay, so what are we doing next for aging? So we're moving forward this work, and this is going to be in collaboration with some folks at UC Santa Barbara, Emily Jacobs and Mary Haggerty. We have a more complete profile, including immersive VR. So here we've got more cognitive tasks, and those were desktop tasks that I was showing you. So we're going to put them in this walking immersive VR, which will help with things like reduced motion sickness. It will help improve proprioception and vestibular cues. So we're having a whole lifespan in addition to focusing on the menopause era. We're also going to go for sort of 18 to 80 and look across the lifespan. So we're hoping to get a whole bunch of people in to test this and see how people do across that lifespan. And then for menopause, we're going to include like a really rich sample of pre, peri, and postmenopausal women, including a hormone assay, Alzheimer's disease risk factors, and so forth, with like next imaging session. And so thinking about factors that are involved. So a lot of this work related to that I've been talking about, thinking about all these like sex hormones and things like that might be involved, is has another relationship. We have a lot of like life hormonal transitions that we go through. And in addition to puberty and menopause, many women also undergo pregnancy. And so I'm going to tell you a little bit that we alluded to earlier that is not necessarily navigation related, but I had a really interesting opportunity to study something important for science. And so we wanted to look at pregnancy. So now about 7 years ago, I was planning pregnancy and was like, you know what, maybe we should take a look at my brain and see what happens while I go through pregnancy. So how does the brain change? And are these changes long lasting? So that's something I think a lot of people are interested in, as something we haven't really studied very much, especially because there's a lot of concern about testing anyone during pregnancy. But as a neuroscientist, it was, I was pretty cool with it. So I scanned myself 26 times before, during, and after pregnancy in a dense sampling collaboration with Emily Jacobs. And so we have a whole bunch of time points here, including up to 2 years postpartum, to look at these changes. And so this is the hormonal changes over time. So a normal menstrual cycle is like this, basically, and then this is like the pregnancy time. So it's quite a lot of hormone changes that are associated here, and this is sort of birth, and they all come crashing down at the end. So you can see that things are happening, and then we can also see how the brain changed. So we found overall really large decreases in gray matter volume throughout the brain, which was across numerous brain networks. Almost everything went in the decrease, about 4 to 8% overall. And so we saw this decline over the trimesters and a little bit of a bounce back, but not that much. So we saw a small postpartum rebound, but again, overall, generally reduction in gray matter volume. Ventricle volume also had a similar change, but in the opposite direction. But it was not all necessarily bad news, and I don't know if that is necessarily bad news. But we also saw that white matter, that sort of communication system, had increases in fiber integrity. And so we saw an increase that peaked sort of between the second and third trimester, and then returned back to baseline. So this nonlinear effect that basically had this little bump here. So if we had just done a pre and post design, so just before and after pregnancy, we would have missed all of this in before, because we would have seen no changes. But so we were able to get that sort of dynamics involved. And really importantly is that we saw some evidence of neuroplasticity in the adult brain. So I think a lot of people say that once you're an adult, things don't really change. You don't really have plasticity, but this does show that your brain can change in different ways. So to summarize our age and sex conclusions, we see some aspects of navigation that start to decline as early as midlife, so 45 to 55. So sorry, folks. But others, which I didn't show you, actually are preserved. So I didn't show you the good news that some things are preserved as well. In midlife notes, we see that white matter is related to performance, but might be different. So it might be different from the relationship to young adults. It might be different depending on sex or menopause status. And we have a lot of upcoming work on aging and Alzheimer's disease that I'm excited to talk about in the future. And then finally, we saw those brain changes associated with pregnancy. So we talked about a whole bunch of things today, and hopefully you've taken something away from our different factors of navigation ability. But again, overall, we really see that all of these things work together to help us become the navigator that we are, right? It's not one single thing, but really a mixture of all of these things that are important, both the things about our brains and bodies, but also the things that we do as well. We get sleep and exercise. That can all be really important. And I just want to highlight that, Alzheimer's disease, these are the sort of things that are about 40% of our risk factor is things we can modify. So like this sort of stuff. And so really that's important as well. So I just want to stress all of that. These work together, and hopefully we can form a sort of nice complete profile to understand what you can do to help reduce your risk, but also what we can then hopefully someday do to help prevent and treat these disorders. So thank you so much for your attention. These are all the folks that I work with here and other places. My funding support in my lab has done such an amazing job. They're so awesome. And thank you again. A little long.
That was awesome. So let's take some questions from the audience. Craig, you want to start? And there are microphones that are making their way around.
Yeah, great talk, Liz. One question is, can you train, if you continue to do these sort of navigational exercises, can you exercise your brain to kind of overcome an inability to navigate well and become better at that? And is that potentially correlated with certain brain conditions that might exist?
Yeah, so I think that's what we all want to find out. And so far, as no one's been super successful with it, but I think there are some possibilities, right? So I think it's figuring out the right thing that's going to work. And it might work for some people, so it might have to be targeted training. But I do think it's possible because I think we can learn a lot of things, right? We can train a lot of things. And I think the other thing is that there's people like around the world, right, who are expert navigators, right? There's the Polynesian Islanders. There's other folks who are able to do some things and navigate really well. So I don't think it's like gone, but we might have to like pull it out somehow, but we don't really know how to do that yet.
Does technology make us lazy? That's only
I think it might. And so that's a question. And so I think the GPS question is that people, who do use their GPS more are worse, but it's helping people do something that they couldn't do otherwise. But we might be, off-putting it, but it might be okay, right? Because we also, have a lot of other things. It frees us up to do other things. So it's not necessarily obviously bad, but I don't think it's good. It's not exactly good, but I don't think it's necessarily bad. But I think one thing that I think we do see is that when people use their GPS and they follow like the turn-by-turn instructions and they're just like, okay, left and right, and they don't do anything else, that's the people who don't do as well at navigating. But people who use their GPS and they sort of like pull up the phone and they look at the map and they say, okay, this is the route. Okay, now I know what to do, right? Those people tend to do better at navigating. And so maybe use your GPS a little bit differently. Maybe don't do the turn by turn. Maybe pull it up onto the, still on the top down view. I think that might be, a little beneficial.
Okay. Simon?
So you learn a new navigation task, and you're tested in the first couple of hours. That's stored in the same place as it's stored 3 days.
That's a great question. Yeah, so what is our long-term retention? So first of all, do people even remember it a couple of days later? And so that's something we've done a little work on. Actually, we have a sleep study with Sarah Mennick's group, and we have seen some things about over a couple of days. And so the storage is, where it's stored is always a question about sort of memory consolidation and how things go. But certainly there's going to be, some folks, again, there's going to be some degradation over time. But again, there's some people who are just like, yeah, I've been in this courtyard 40 years ago and I remember it, right? So there might be people who can pull these things up and others that can't. But so where it's stored and how is, I think, part of the question that we still don't know yet.
So, do you need me to use a microphone?
Yeah,
I have a couple of questions. The first one is, you gave us 2 people at the beginning of the lecture and asked us, which one to guess. I'm now guessing it was the young man who did better from the data that you just showed us. And then I wanted to ask, because I don't know as much as you do about the brain, but does data retrieval locations in the brain affect the performance here? And I'm asking that partially because I'm thinking about the lecture we just saw this last weekend with Mary Lou Henner and her amazing ability to retrieve data from her brain. So how could we use her particular skill set to explain some of the data that you've seen?
Yeah, so 2 things. So the first examples that I showed, I should try to mix it up, but I think the young man probably was more likely to do better. But they're all not. They were, some of the factors were moved around. So, But then thinking about questions about these sort of people who do really well. So if people don't know this, there was a lecture over the weekend on Mary Lou Henner, who's an actress, and she's perhaps most well known for her highly superior autobiographical memory. So if you give her a date, February 12th, 1987, she'll be like, oh, that was a Tuesday, and this is what I had for lunch, and this is what I was wearing, and this is who I was with, and what I did, right? So she can remember every day of her life basically perfectly. There might be people who do that, who are similar to that with navigation with some things, like who might have similar things about like being able to recognize places that they've been or routes that they know. So that's possible. I'm not sure if it's been studied. Usually we think about it in this sort of autobiographical thing, these like events, but it could also be that there's people who could do that with places. I don't know of anyone though, but I think it's possible. So I think being able to then pull that out, I think, there is this huge relationship between memory and navigation, and that's why they're so overtwining, right? So everything that we do, all of our autobiographical memories happen at a place and at a time. And so we have, location memories for things that happen to us, right? We can remember this room and seeing this talk, which happens at a time, but it also happens in a place, right? And you can maybe remember who you're sitting next to. So there's all those different things, all those details go into it, and they're all part of it. And so they're super intertwined. It's hard to separate it out. And so that's one reason we kind of look at different aspects. And so I tend to study the space more, but I'm also interested in time and other things too, because I think they're all important when we put this together into a sort of memory system.
So you mentioned that white matter integrity matters. Is it known if individuals who suffer from demyelinating diseases, for example, are worse at spatial navigation?
I don't know. That's an interesting question. So I don't know if we have tested people on that. So I think it's an interesting question.
Hey, okay.
I was really interested in your finding about the dynamic functional networks, and I have two related questions to that. One is, what is the anatomical part? So is it like one neuron, or is it like a network of neurons? And then the second question is, you talked about how it's like when they can swap back and forth, that's predictive of learning. But what about if it's really stable? Is that predictive of a different type of skill? Can you comment a little bit
that's an
about
interesting question.
stability
Yeah.
of those networks?
Yeah, so we're looking at is like the region level. So in this case, we broke the brain up into about 200 parcellations, which are sort of, I don't know, like this big, but they're sort of like little brain regions, which are going to have like hundreds of thousands of neurons in them. So they're all going to have a lot of neurons in each of these, but they're sort of like functional units, like the precuneus or something might be broken up into like 3 or 4 different parcellations. So that's the kind of level that we're working on. With the human brain, we don't ever go to the single neuron level, but we can see, regions. But the stability question is interesting, right? So, I think sometimes we're like, oh, stable things are better. Just, because it's like less wild. And so that's what we're trying to understand about with this situation. So we saw that, changes are good, but stability could be useful for other things, right? And that maybe that's useful for either like retrieval, right? So this is the learning process. Maybe it's useful for retrieval. Maybe it's useful for well-learned kinds of things. And I don't just know, so I'm just like kind of making things up right now, but there probably are aspects of stability that are going to be helpful. So I'm sure that there's like a balance, right, for all of these things between stability for certain things and flexibility for others.
Okay. One last question.
Comparing the structure imaging and the functional collection, which one has a stronger correlation with the prediction of performance?
So between structure and function?
Yes.
So we don't know as much yet. So in terms of like the machine learning, we've only put the structure in. So we haven't included any function in that at all yet. And so I think when we include function, that could potentially be even more helpful. I think that function could be very useful because you can start to look across the brain and you can start to look at multiple regions. And so again, one thing I think that's something that we work a lot on is not just focusing on a single aspect. And so it's not just all the hippocampus, it's not just all retrosplenial cortex, but we're really looking at the whole thing. So we might look at, all of the places together to see their cortical thickness or other things, or how they interact together. So my gut would be that function is going to be more useful in the end, but we can still see that something about the structure is telling us something.
Okay, so let me just say that I want to thank you, Liz, for giving not only a wonderful talk, but really breaking down a topic that affects all of us on a daily basis and talking about all the different biological factors that impact it. And she was a little worried that she wasn't distinguished enough to give this talk, but I think her data really prove otherwise. So let's give her a warm round of applause. And just a little
Thank you so much.
gift to thank you. And now I'm going to invite all of us to spatially navigate to the reception so we can enjoy some fun times. And Liz will be available to answer any of your other questions. So thank you all.